Hello, and welcome to She Leads Biotech, the podcast that celebrates women leaders in the bioscience space. I'm your host, Sharron Cox, CEO and Managing Director of CNA International, based right here in New York. And each week I'll be talking to women leaders about their work, their triumphs and challenges, providing invaluable insight into how women can unlock their superpowers and succeed on their own terms. So whether you're navigating your own career path in bioscience or you're passionate about science and innovation, I hope you're empowered and inspired by each episode. From a 7th grade spark in science class to shaping the future of cell therapy, today's guest has never been afraid to step into the niche corners of research where few others dared to go. Armed with a PhD in microbiology and immunology, she's built her career on asking questions nobody else was asking. Through her successful consultancy, CSM Consulting, she guides biotech and cell therapy companies and collection centers on how to get donor materials right from the very start. She's passionate about education. She's built a following with her videos, and she's on a mission to make sure the industry never overlooks the starting materials that can make or break a therapy. So please welcome the Leukopak Lady, Jennifer Chain. Jenny, hi.
Hi, how are you?
I'm very well indeed, thank you. I think it's still morning for you, isn't it? Afternoon for me. Where am I speaking to you from?
So, it's 11 o'clock here, and I am in Norman, Oklahoma.
Whenever I hear that word, I just want to burst into song.
Oklahoma?
Every time people say Oklahoma to me, I almost go, no, that's not an appropriate time to do it, Sharron. Stop it. And it's not a state I've ever been to, I have to say.
Okay.
Are you a born and bred Oklahomian, or however that is?
Oklahoman? No, I'm actually from West Texas. I'm from Amarillo, Texas, which is in the Panhandle. But I went to college and grad school here in Oklahoma, and I met my husband here, and then we moved away to Colorado for 8 years, and then we moved back.
Okay, all right. So you've been around the Midwest a bit.
Yeah.
Okay, yeah. All right.
So.
And I know from your profile, gosh, you've done so much. You know, you're a scientist, you're a business owner, you've been a C-suite executive, and obviously I want to dig into all of that. But how did it start for you? What sent you on your initial journey?
Just in science in general?
Yeah. Oh, yeah.
And it really honestly started in 7th grade. I was probably interested in science, but I loved life science in 7th grade. And my teacher came up to me one time and he said, "You should be a doctor." And I was like, okay. And so that kind of got the wheels turning. And throughout high school, though, I started to become more and more panicked about being a doctor. So when I got to college, I talked with my advisors and stuff, and they're like, "No, I think you would be better suited for a research career or a lab career." I did summer research that led into a job. And then I also did another competitive summer internship doing research, and that helped me know that for sure I wanted to get a PhD and that for sure I wanted to study the immune system. Now, all of a sudden, the immune system was so interesting to me whenever I did it in practice. In the class, I couldn't get it. It was boring. It was hard to understand. But once I got my hands into it, I loved it. And so I went to grad school and studied immunology. I studied how the T cells, which is part of the immune system, how those developed. And there's a rare type of T cell called a gamma delta T cell. And I got really interested in that. And so I went to do my postdoc studying gamma delta T cells. And then from there, I had some funding that didn't lead to a great result. Like the project didn't work out like we planned. And so, that can really derail your research career. And so I explored other avenues. And also I wasn't really sure I wanted to be a professor by then anyway. You go into grad school thinking you wanted to be a professor. And then I wanted to be able to apply more of what I learned to problem solving and practical applications. And so I got a job in industry, working at a biotech startup, doing diagnostic testing and diagnostic testing development, like R&D. And then I did lots of troubleshooting during my graduate work and my postdoc work, building new procedures and troubleshooting them. I just decided to offer that as a a help to others. And so I started my first consulting firm in 2016. And I helped train staff. I did experiments. I did all kinds of things. I learned new things. I grew a lot as a consultant. And that got me connected to the blood center in Oklahoma. They needed help kind of breaking into the cell therapy world. And they didn't really have that expertise in-house, that scientific expertise. There's lots of great expertise at the blood center. They've got medical and laboratory expertise, but that research and science background is not there for most blood centers.
Because it was still pretty early then,
Yeah.
was it? I mean, even when you did your PhD, and as you were saying there, talking about, gamma T cells and things like that, that in my head is still, gamma rays, and that's Incredible Hulk, and it was all still very sci-fi-ish at that time. How did you know that this was going to be the future? How did because I imagine there was a lot of really, you're going to be doing this. How was it while you were going through that process?
So one of my motivations, I've learned this along the way, is to do something that not every single person out there is doing. Right? I didn't want to do cancer research because everybody did cancer research. That was my early thinking. So gamma delta cells were a very small niche, and there was a very small community. Right. And so that has seemed to happen with each stage of my career, is where can I find the thing that nobody's doing or that I can add value to in a new way. And so that has eventually got me to where I am with leukopaks and starting materials. There's certainly people out there that know a lot about starting materials and they're experts, but there's so few, and so few people are thinking about starting materials in the grand scheme of manufacturing. A CMC expert just receives the starting material and starts manufacturing, and they don't realize that there's this whole world that comes ahead of that first step in manufacturing, where you have to care for the donor and you have to recruit them and you have to test them. You have to get a good product collected. You have to find a good donor. The choice of that donor is going to affect the manufacturing outcome, and it's going to affect the clinical outcome. Right. And I think that is a concept that still hasn't filtered all the way out into the cell therapy community, is that downstream successes start with the choice of the donor. And so I wanted to be somebody that cared about that donor, that cared about that quality of the collected product, the starting material.
Right. And that was actually one of the questions, and you may well have answered it, but my question was, you seem to have carved out this niche in the starting materials area. And it's an area that most people outside of cell therapy don't really take a huge amount of they don't think about it, I guess. What is it that you wish more biotech executives understood about the strategic importance of starting material?
Yeah, that you need to have a plan at your company to say what's important to us, and how do we figure out which donors are the right donors to meet that important thing that you've decided. So, common things are, the donor can't have chronic inflammation, because the chemical messengers, the cytokines that are in the blood and in the soup during chronic inflammation, affect how the T cells function, which will affect how they're manufactured, which will affect how they fight cancer in the patient that those cells eventually get into. And so what are the factors that affect the function of your therapy, and how do we figure out how to ask the donor the right questions, do the right testing on the donor and the collected material to say, okay, out of 100 donors, there's only 15, or there's 25, or there's 50, whatever it ends up being. There's only a small number that are actually right, that will actually give a good outcome downstream. And so I wish more people thought about that. And they don't necessarily have to do all that work. They have to define what's important, but then they have to find a collection center or somebody like me that is willing to help them ask the right questions of the collection center. How can you get to that point where now you're saying, okay, these are the questions to ask, and this is the testing to do. And so we can narrow down which donors to even collect our products from that we'll use in manufacturing.
What's the benefit to a biotech executive of doing all of this? Because I imagine that at first it's like, oh, that's a bit of a faff. Look, we've got 100 people here who are ready to, they're ready to go. Can we just get on with that?
Right.
But what would you say to them is the benefit of just hold fire. And look, at the
Mhm.
end of the day, if we've only got 15, here's why it will still be a good.
Yeah. Yeah, because I think if you continually have failures in clinical outcomes and you don't figure out why, you continually have failures in manufacturing outcomes and you don't figure out why, eventually nobody's going to approve those therapies and nobody's going to give you funding to make those therapies. And so I think the downstream consequences of not starting with the best quality starting material is going to be that this side goes away. Allogeneic therapies, these are donor derived therapies that go from a healthy donor to a patient, a sick patient. Those are still new. They're behind the self derived therapies, the autologous therapies, because there's so many other factors to consider. And so if you add one more variable to that, it's going to make it harder and harder for these therapies to look like they're working.
Right.
For the numbers, the clinical outcomes, to say, a lot of patients, 80% of patients, 60% of patients, 90, whatever, had good clinical outcomes. And when it's 20, 30, 40, one of the reasons could be that the starting material was inconsistent. You're getting some good starting material and some not good starting material, but you didn't think to ask those questions from the beginning. And so asking those questions from the beginning can make your therapy versus break your therapy, is what I think.
Yeah. So it's better to have a 95% efficacy on 15 patients than a 40% efficacy on 150 patients.
When you're talking about 15, it's really donors. So these are healthy donors.
Right.
And the ratio of donor to patient is higher. It's not the self
I'm with you.
donation. So it all depends on the therapy, but one collected starting material from one donor could make 10 doses, a dozen doses, 50 doses, maybe even 100 doses. And so when you find those good donors, you want regular
collections. You want to be able to keep going back.
Yeah. And then that becomes an issue with the collection center. How are they with their donor stewardship? Are they maintaining good relationships? Are they treating them well? Are they comfortable? Are they minimizing their time on the instrument and their side effects? There's all these things to consider in donor stewardship once you find those donors. Now, if you're going to collect from 100 donors and you don't really care whether they come back again, you might treat them differently. Right. And if you know you're going to need those donors to come back once every 8 weeks or 4 weeks or 12 weeks or something like that, then you maintain that relationship.
Right. And yes, thank you for that, because that's definitely schooled me. I'm thinking in terms of trials and patients, and in your space, you're thinking about donors, like blood donors, where you would go and give certain blood to keep the banks topped up for as and when they're needed.
Yeah, exactly.
That makes much more sense to me now. And I want to ask, well, while you were studying all of this and you spent your time in academia as a scientist, how was it for women during that time? Was it very, I imagine that there weren't that many people in the space at that time. Was it very female focused or equal?
This is surprising when I look back. I didn't really think about this very much at the time, but I had female mentors. I can think of 3 off the top of my head that were very prominent in the department. Now, maybe 4 or 5 that were very prominent in the department, that had been scientists for 20, 30, 40 years by the time I interacted with them. And so there's actually in immunology, or at least immunology in Oklahoma, there are a lot of great women that trained me, that trained all the other people around me. And so I never felt like there was a shortage of women in my small field or my niche field. Engineering fields and some other fields, there might be more of a disparity, and maybe other parts of the country there might be more of a disparity. But from my perspective, there were a lot of great female mentors that I had access to.
Okay.
And I can thank for my success, for sure.
You went into industry. You did work with the Oklahoma Blood Bank. Where did you go from there? How long was it before you did your next consulting business, which I really want to kind of focus on? But finish the journey for me that got us there.
So at the blood center, the first few years I was there, I wore a lot of hats, and I helped the cell therapy team develop cryopreservation protocols and cell isolation protocols and flow cytometry and some other things, and help with training. And then I worked on developing some new products. And then all of a sudden in 2021, I had the chance to lead the collection program for healthy donors. So we had already started it, and I'd been moderately involved, but not heavily involved. And so now all of a sudden I'm the lead of that division, and nobody was there to help me. The person who was doing it left, and so I was trial by fire. I had to figure it out. I was recruiting donors. I was interacting with the clients. I was trying to understand their needs. What are their needs in donor qualification and testing and collection and processing and shipping and labeling and all of that stuff. And I was doing most of that on my own. I had some help with phlebotomy. I eventually had a really, really amazing person helping me with logistics, and took over some of that client engagement. And I had eventually hired a recruiter, but at first I was doing all of that. And so my analytical mind, I was problem solving. I was figuring out how to make it better, where there are kinks, how to interact with the donors. And it's different than with blood donors. It's a much different animal. I couldn't learn that much from blood recruiting. I had to kind of figure out how to reach our donors in my department separately. And so after a couple of years of just being immersed in that, I just fell in love with it. I'm like, this is my calling now. This is what I want to do, because it brings in that consulting side, because I'm consulting with the external clients and project managing. What do you need? How can I make it work? And then I'm consulting with the internal teams. This is how we're going to make it work. How can we do this? I really just developed a passion. And then all of a sudden it was gone. I couldn't do that job anymore. And I was devastated, right? I was going through a mourning process. How do I keep doing this work without a company that I'm working at?
Right.
And so that's where I came up with the idea for CSM Consulting, and CSM stands for Cellular Starting Materials. And I wanted to focus on how to help other centers start or grow. Some centers had already started, but they wanted help with marketing or developing donor databases. And some didn't even have an instrument, but they wanted to get a program started. So let's start from the beginning. And so I was able to help. So far in the last year and a half, I've helped 6 nonprofit collection centers and 2 for-profit centers start or grow at some part of their program. And one of the things that I also did in 2024 and early 2025 is I tried to start my own collection center with 4 other people. And there were some roadblocks. There was some things that ended up not going well. We were trying to buy an existing center. We were trying to raise money, and those things ended up not working out. But during that time, I'm just immersed in, how do I start this center? How do we make it work? How do we qualify donors that meet the needs of the industry? How do we advance the collection science, and how do we do cryopreservation? So it's really focused on how to serve the cell therapy community based on my experiences so far. And so once that stopped, it kind of almost stopped all of a sudden. I didn't have that company that I was trying to start. And then all of the other companies that I was helping at that point, I had finished the work that I was contracted to do. And so there were a few months there where I wasn't thinking about starting materials. I wasn't thinking about leukopaks. And I was sad. I'm not motivated.
That's very much, that's your DNA, isn't it? That's, yeah.
Right. And I had figured that out by then. And then this idea popped into my head. Maybe I should just start talking about it. I'd already been writing some articles. Maybe I should start just putting out some videos. Other people were doing something similar. So maybe if I just talk about what I know, nobody's paying me to do it. I'm not really trying to impress anybody, except I want to be educational. So, I want to do it in a way that is helpful, but I kind of had some imposter syndrome moments. What if nobody likes it? What if I can't, follow through? And so I had, laid out a 14-week plan over the summer. And I finally just thought, what, it doesn't matter. If nobody watches it, I still enjoy doing it. I'm going to write the scripts. I'm going to film them. And that way I'm writing and I'm talking about leukopaks and cellular starting materials and donors and stuff like that. And it lifted my spirits. It made me so happy. And I followed through with it all 14 weeks, even whenever I had to figure out new topics that I hadn't planned out, So I'm like, hey, I did this. I followed through. And I like doing this, so I'm planning another season 2, where I'm going to probably do a lot of similar things, but a couple of new things, like maybe some AI incorporation and some fun stuff like the donor experience, and follow a donor through their process and follow a leukopak through its process to become a therapy. So a couple of cool things like that.
I love the videos. I mean, I actively look for them.
Thanks.
I have to confess that I'm a recruiter.
Mm-hmm.
I'm not a scientist, so I'm not an
Yep.
expert. So a lot of that, it does tend to go over my head. And I rely very heavily on experts like you to school me
Mm-hmm.
in those pertinent areas. But and I've seen that the most recent one you did was almost like a recap of season
Yeah.
1, right? And
Yeah.
I really highly would recommend it, people who would find this interesting and who'd understand it to go back and start that. And 2 questions I want to ask you about that. A, where can people go to
Yep.
see the videos, and who would be your audience? Who would benefit by watching those videos? And they may be out there not even knowing that they would benefit by watching your videos. So who would that be?
Yeah, so if you want to go on LinkedIn, you can search the hashtag Leukopak Lady, all one word, and that should pull all of them up because they were all posted on LinkedIn. And I also eventually posted them on YouTube, the Leukopak Lady.
Okay. And who would want to watch it? Who would benefit by watching these?
So the audience in mind were people that were at least somewhat in the know. You know, they understood the language. Well, yeah, they somewhat understood the language. And that was something I went back and forth on. You know, how basic do I want to make it? I wanted people that were developing therapies to know that there is somebody talking about the collections, or somebody thinking about how that product gets to them. Collection centers I have tips in there on how to, standardize the collection process using a calculator, a couple of other suggestions that I have in there. And so collection centers might benefit if they're looking for a way to try and meet the developer's needs. What are they looking for? How can they better standardize? And so I think both those fields, the collectors and the developers, that's how I think of them, could benefit because it has information that they can learn from. There's a big regulatory block in there. And really it's me kind of almost thinking out loud because I'm trying to figure it out as I go. And it's very confusing, but I distilled it down to as simple as I could so that somebody at a collection center or even a developer who's working on their IND, what do they need to know about the regulations that are specifically about the collected material?
Okay. And actually that kind of brings me on to my next question, because I know, and I would imagine that all of the work that you did when you were looking at setting up your own collection center, you would have gone through each of what are the challenges we're going to face, what are the problems we're going to hit, etc. So that's a big part of your consulting business. And you've helped collection centers build or grow out the leukopak divisions, right? What would you say is the most overlooked factor when setting up a successful program? I mean, is it the donors, the operations? Is it the sales, marketing? What are the big things that somebody should look out for if they're having challenges with their center?
Yeah. So I think my perspective is there's a race to the bottom. There's so many collectors. At last I counted, in the US alone, there's 34 collection centers that are blood centers or for-profit centers. That doesn't count hospitals or brokers. And so they all want to have the cheapest leukopak. The cheapest leukopak means that the donors are random and that they're not, it doesn't mean that the leukopak is of bad quality. It just means that there's not a lot of donor qualification. There's not a lot of processing. And so there's so much donor testing that should be done. It's working closely with the developers. What kind of testing and qualifications do they need? Operations, I feel like almost anybody could do the operations. Blood centers have their infrastructure set up, and so it makes sense to set up the leukopak operations, and hospitals have it. So setting up the operations is challenging, but not the most challenging part. I would say understanding the industry need, the developer's needs. What do they need in donors, collections, and processing? And understanding the regulatory aspects.
Right.
And so the regulatory aspects were designed for autologous treatments, which are patient-derived. And then they were modified slightly for healthy donor-derived direct transplantation. So like stem cell transplants. And so all these therapies that go through manufacturing, the regulatory aspects are not well defined. And so it's really hard to navigate. And it's almost like you have to fit your program, your collection program, into a box that it doesn't really fit into, regulatory box that it doesn't quite fit into.
Right.
And so it's really hard to navigate the regulations. And I hope that someday that the gaps in the regulations are filled in.
Okay. And you talk about these autoglass therapies, and I know you've mapped out the kind of vein to vein value chain of these. What did that process reveal about the bottlenecks, where the biggest bottlenecks are?
That's actually a really good question. And that goes into manufacturing. And I don't feel like I'm an expert in manufacturing, but I have seen where like the decentralized manufacturing model, where you're going to collect and manufacture and treat all within a very close geographical radius. That's such a great idea. It's going to make patients get their treatments faster, but the quality control and the talent, where are you going to find all these talented manufacturing scientists? Where are they going to come from to each individual location? How are you going to make sure that location 1 and location 2 produce the same quality product? So those are some of the bottlenecks, is how are you going to train all of these staff, all the systems, the electronic systems to monitor all of these things are not quite there. And so that revealed that there's so much in between, donor to patient or patient to patient. All that stuff in between is not as well defined in a decentralized model as it is for a centralized model. The centralized model, there's issues with transport, and there's issues with both models, but that one is better figured out right now. And so getting to that closer to the patient manufacturing is going to be a challenge. And I think it's going to take a long time. And I think it's going to take the development of a lot of systems and a lot of monitoring. And it's going to take a development of the talent workforce in cell therapy specifically.
Yeah. You touched on it briefly, and I may be totally off pat with it, but you talked about that the centers, collection centers, want the kind of cheapest leukopak.
Yeah.
How is that education process to the collection centers, in the fact of, but cheaper doesn't mean better. Cheaper doesn't, how can that be explained, or is it explained that, the work that goes in, just as we've been talking about with the starting materials, so that you've got a much better qualified number of donors that might be more expensive to deliver, because obviously of the amount of work going and weeding out all the bits that aren't any good, but at the end of the day is going to make that huge difference in the quality of what they're working with. How challenging is that to get across to collection centers?
Yeah, I think the challenge is, because I've had people say that, donor qualification is too expensive, we're not going to do that part. They just flat out say it, because the instruments are expensive, and the involvement is more involvement in labor and everything. And so the cell therapy company that wants to buy the leukopak, they're going to have to communicate to the collection center that we need more in donor qualification. And the cell therapy company can buy the instrument and do the qualification themselves, and then just tell the collection center what they want. And, that's one way to do it. But the collection center can be proactive and buy the instruments that they need to do the qualification. They can bring in the expertise to do, cell assays, proliferation assays and stuff like that to get some of that started. So the more the collection center can help the developer, the better they look to the developer. And, some of these leukopaks go for, somebody told me the other day, $40,000. And so we're talking $10,000, $20,000, $40,000 when the blood centers are charging much less than that. And so the industry will pay for it. They just need to offer exactly what the developer needs. And the more they dive into the industry to figure out what the developer needs, the more proactive they can be. And if not, then the developer is going to tell them exactly what they want, and then still expect something cheaper because they've done all the work. And so I think the education process starts with the developer, and the more the collector works with the developer to understand what they need, the more proactive they can be.
Okay. All right. So what's coming up for you? Is there anything you're working on at the moment that you can tell us about, or what are your future plans?
Yeah, so I in the end of September, I went to CAR TCR for the first time, and I was part of a panel with the company execs, and I have the panel title in front of me: Donor Screening and Point of Collection Characterization to Ensure Leukopak Quality and Cell Therapy Development and Manufacturing Success. So that's a huge
Okay.
mouthful,
That's a mouthful.
but it is a mouthful, but the title is so perfect because it covers everything that I've been talking about, that the
Right.
quality of the starting material is important to the manufacturing success and even the clinical outcomes.
Okay.
And so, that panel was focused on talking about how to get those products, the starting materials, better qualified to
Okay.
ensure that success. And so
Okay.
I was glad to be a part of that. And then coming up, I'm going to be at AABB, which is the Association for the Advancement of Blood and Biotherapies. And so it's traditionally a blood conference, but in the last, 10, 12, maybe 15 years, they've started to focus more on cell therapies and biotherapies. And so I'm heavily involved with that organization on that side. I'm on committees and stuff like that. So I'm going to participate in some of the activities there.
Excellent. And where is that? Where is the conference in October? Where is it being
Is
held?
in San Diego. San Diego.
I love San Diego. I'm really looking forward to going there in February for the Advanced Therapies Week again. Last one was in Dallas. We met there, right? In Dallas.
Yeah, that's where you and I met.
Yeah,
And
not a lover of Dallas, I've got to be honest.
yeah,
It's another story.
I'm from Texas, so I enjoyed it, but I'm looking forward to going to San Diego as well.
So anybody who missed Jennifer at the September event, you can catch her again in San Diego for the October event, but also reach out to the Leukopak Lady, because what she doesn't know about cell therapy probably isn't worth knowing. I feel so educated, Jenny, I have to say, and I totally get it now, and I understand the processes. So thank you very much indeed for doing that for me. Closing question for you. Obviously, you've been doing this for many, many, many years, and there are challenges at the moment for in the industry, B, for scientists coming through, questioning if they'll even get research money to do the research. What would your advice be to a keen scientist looking for an opportunity in the cell and gene therapy space, but not sure? What would you tell them?
Yeah, there's lots of opportunities to get involved and learn. So there's AABV, there's ISCT, there's ARM, there's tons of committees, there's tons of organizations to learn. So if you're wanting to get involved in cell therapy, that's really what I did. Is I joined committees and I learned from my peers. I learned from other people talking, and there's so much to do in cell therapy. I can't predict what's going to happen with funding and with, things like that, but I am optimistic that results will drive the field. So good results will gain approvals and will get funding. And so I think there's a future in cell therapy for somebody that wants to get in it. And my personal advice would be to find some place that you're passionate about, where you fit in, that you can make a difference. And so there's a lot of noise out there, and there's a lot of people doing great things. You got to find what you're good at, where you can contribute. And if it's something brand new, then that's great. And then, the field grows.
Okay.
And so
also find those mentors that like you did.
Oh yeah. Yeah,
that
absolutely.
can maybe help you navigate the paths more effectively.
Yeah, exactly.
Excellent. Well, listen, I'm very conscious of your time.
Thank you.
I really appreciate you taking the time out to chat with me today. And as I say, I feel so educated. It's not even funny. I'm going to be talking about cell therapy and leukopaks probably all weekend now. Boring life out of anybody I know, but I appreciate you educating me in that and for telling your story. So I wish you every success with your consulting business and what you continue to do. You have so much knowledge that people can gather. So keep telling your story. I'll keep watching the videos, and we'll make sure the algorithm starts moving it out so that more people can see it. But have a fantastic weekend, Jenny. Thank you so much for your time, and hopefully we'll meet again soon.
Thank you so much for having me. I'll see you.
Pleasure. Take care. Well, that's it for today's episode. Thank you so much for listening. She Leads Biotech is brought to you by CNA International USA. a woman-owned boutique search firm based in New York. If you're looking to hire talent for your company or want support in achieving your career goals, please reach out to me at contact@cnaint.com. I hope you enjoyed this conversation. And remember, the success of every woman should be an inspiration to another. So don't be afraid to share yours. Thanks again for listening. Until next time.